Breath. I want to welcome all of you to today's K Special Interest Group meeting. These are monthly meetings primarily focused on clinical investigators in various aspects of your careers. With Naomi Lubin, wave your hand at Children's. We organize a series of workshops really focusing on helping new investigators create their research projects and be successful at both GW and Children's National. Today, we have a pretty focused topic, one I think that will cross over to other kinds of clinical trials, but we have three wonderful experts to talk about some of the challenges and successes in accruing to cancer clinical trials. Dr. Chalasani tells us she's running late in clinic, so I think we'll get started and allow her to come in just a couple of minutes when she's available. What we thought we would do is start out with very short presentations from our three speakers today and really commit most of the time to your questions. So I would encourage the audience to either put questions in the chat or to raise your hand, and we'll try to monitor those as we go through. So what I think we'll do, depending on when Dr. Chalasani comes in, is start with Julie Bowman, who's going to be talking about why are we even talking about this? Why do we care about clinical accruals to clinical trials? Why is that important to the Cancer Center and to investigators? We'll then move on to Dr. Arang Kim, who's going to talk about some of the unique challenges with pediatric cancer clinical trials. Again, some of the challenges and successes in early phase therapeutic trials. And then when Dr. Chalasani comes in, she's going to talk a little bit more about from the physician perspective, how do you learn about feasibility for your trial? How do you select trials for your patients? Some of the definitions and enrollments and so forth. So I think each speaker has maybe 5 minutes or a few minutes to talk about their subject, and then we'll open it up for questions. So Dr. Bowman, I invite you to start out, and I think Pesha's going to show your slides.
Thanks, Pesha. Next slide. So I'm talking about clinical trials in the context of building a cancer center that is NCI designated. And I like to remember the reason why, the why behind clinical research, which is to drive transformational research, personalized therapy, family-centered care, and cancer policy in the nation's capital. Creating a cancer-free world, I really described clinical trials as the last mile. This is where our discoveries touch our patients, and being at the bedside to be a part of the transformation of the field of cancer has been the joy of my career. Next slide. So what is an NCI designated cancer center? About 50 years ago, President Nixon, the National Cancer Act, which established the first 15 NCI designated cancer center. And this is a grant. It's a grant from the National Cancer Institute. The Cancer Center director is the principal investigator of that grant, and it recognizes rigorous standards for transdisciplinary state-of-the-art research that's focused on prevention, diagnosis, and treatment within a specific dedicated catchment area in which we can demonstrate our impact. Right now, there are approximately 72 NCI designated cancer centers, and we aim to become a new one. Next slide. So I'm going to spend very brief on this, but I just wanted to point out that part of the Cancer Center grant, they review our science, they review our impact, but what the Cancer Center Support Grant funds is infrastructure. It's a grant that funds cores. And the clinical trial piece of that grant stands out as approximately 10% of the average grant that supports that clinical research infrastructure. Next slide. In a cancer center, there are two major components related to clinical trials that are scored. One of them is the clinical protocol and data management system. This is essentially the clinical trials office, or our CTO, where accrual to clinical trials is the primary metric. And then there's the protocol review and monitoring system, which includes scientific review, monitoring, data safety and management, where trial data is the primary metric. And the one thing that I wanted to point out that's unique about clinical trials is that there is a two-stage scientific review that is to occur in a cancer center and to which we are accountable. And that is first a scientific and feasibility review by the disease team that's curating a portfolio, say, in GI cancers. And then second, our actual scientific review committee that reviews all of those things and follows the trial along. Next slide. So building an infrastructure has been my major priority as the Cancer Center director here over the last 2 years. And here are adult interventional accrual trends. The other things that we're tracking about trials are the kind of studies there are. Are they externally peer reviewed? In other words, they're grant funded. A PI has a grant and it is actually funding that clinical trial versus industry, institutional, or national cooperative group studies. And a key metric, this is a loose metric, but everybody, we're not supposed to talk about it, but it's always what we're being judged by, is that when we reach a threshold somewhere between 10 and 12% of accrual to interventional treatment trials, where the denominator is our newly registered analytic cases, that is when we get to the point where our clinical trials are sufficient for NCI designation for submitting our first P30. And so here, that would be somewhere on the order of 150 patients go on to clinical trials. Next slide. So I have set these targets as the Cancer Center director. I set the target each year for what I'm hoping that we can accrue to clinical trials. In 2023, we set that target at 30, and we exceeded it with this nice upward slope at 35. And as you can see in the lower right hand, almost 60% of patients who go on clinical trials here are Black. You can also see in the upper right-hand corner, my goals, which I think we're on track for as we build infrastructure to accruals to therapeutic and interventional trials to have us ready for NCI submission in approximately 2025 to 2026. Next slide. There are a few tools in the Cancer Center. I think Pavani may be getting more into feasibility, so I just wanted to highlight, I think Lexio Barr is on the call. She's the project manager, can do a significant amount of help with investigator-initiated trials once a good draft of a protocol has been presented. We have Microsoft Word templates on our clinical trials office webpage. There's a protocol builder available for through the School of Medicine and Health Sciences. In terms of ideas and generating ways of preparing and developing IITs, I think the scientific programs of the Cancer Center, particularly the Clinical and Translational Oncology program, are a very good source. So attending our research program meetings and scientific seminars, as well as individual meetings with GWCC scientists who are very accessible, particularly when you're considering a trial that looks at mechanism or molecular endpoints. And for biostatistics, one of my lessons learned is collaborate early, collaborate often with biostatisticians, and don't think that you can do that on your own by looking up a phase 2 chart in a book. I'm going to leave the feasibility to others, and that's the end of my presentation.
Fantastic. Thank you. That's a lovely overview. Can I ask one question immediately, which is, do pediatric accruals count toward the CCSG?
That's a complicated question, and it depends upon whether we go forth as one cancer center or ultimately a consortium.
Gotcha.
And that's still
that's a complicated issue.
Yeah, that's still a complicated issue, but children's is very healthy with regard to clinical trial accruals.
Fantastic. Thank you. Okay, Dr. Kim, and I see Dr. Chalasani has joined us. Dr. Kim, would you like to give us a little bit more detail? Yeah. What's unique about pediatrics?
All right, and I can just share my slides here. Okay. All right, so I'm going to be talking about clinical trials and accrual from a pediatric oncology perspective, as well as an early phase clinical trials perspective, which is primarily what I do in terms of my clinical research. So some of the unique characteristics in pediatric oncology is that pediatric cancer is, what we would consider a rare disease. So when we think about, I do mostly solid tumors and I do mostly sarcomas. And when I think about rhabdomyosarcoma, which is the most common soft tissue cancer in pediatrics. We see about 400 cases per year across the United States. So peds cancer is rare, and as being rare, most pediatric cancers require treatment at a tertiary care center. And that being said, most pediatric oncology centers belong to major consortia networks. And primarily a very large one is Children's Oncology Group, or the COG, particularly because we have to work in a cooperative setting to make any real meaningful changes for, in particularly, our new and upfront studies for our patients. And about 90% of all pediatric cancer patients in the United States are treated at what would be considered a COG institution. And about 50%, although you'll see, a lot of wide variances in the literature, anywhere from 20 to 75% are treated on active clinical studies. Here at Children's, we have about 145 active clinical oncology research studies open, and we have about out of those, probably about 40% of patients are treated on clinical studies, but that I didn't tease out the denominator because not all pediatric patients that enroll are eligible for studies. And if we think about our registry and biorepository studies, the number is very higher. Like our lymphoma and solid tumor patients, about 90 to 95% of them are enrolled on biorepository or registry studies. So when you have a new cancer diagnosis, the likely is if there's a trial open, it's been vetted, we've gone through it through a cooperative setting, and if the patient is eligible with minimal requirements because they're a new diagnosis cancer patient, they're often treated on a clinical trial. And so the referral base is from our pediatricians across the area, and they come to Children's because we are the pediatric oncology center in this area. I think what changes a little bit when we think about it in terms of referral is when we think about new diagnosis versus the recurrent and refractory setting. And in the recurrent and refractory setting, we often think about early phase studies. So phase 1 and 2 studies, the first comers, these are first in patients, but in peds, it's often first in children, which is often after adult studies. But what these are typically novel therapeutics that are offered at one or a few centers across the country. And in order to do stuff, we need to have a specialized center. So not every institution can open and develop early phase studies because you need an investigational drug services, you need a clinical research staff, you need the infrastructure, sample processing centers, storage facilities. But these often are where we get referrals, not from the general population, but really from other pediatric oncology centers. And the reason for these referrals are primarily for patients to gain access to new agents and trials. And so, that's partly and some of these are derived from families themselves, and some of these are derived from our pediatric oncology colleagues. So how does this affect in terms of recruitment and accrual, and how do we think about it, especially, as a clinical researcher when we develop our trials? So one of the things that I thought about was why do we need to separate even pediatric and adult phase 1 studies? And could we integrate them more? And I think there are times we do have to differentiate them, but there are times I think we can integrate them more in a setting. Obviously, pediatric and adult cancers have different histologies. So if we had a study that was specifically open for non-small lung cancer, then it probably wouldn't make sense to open in a pediatric center. But there are now, with molecular signatures that are happening, that are based on agnostic factors. So if you have a TRAC fusion, you can have a TRAC fusion ovarian cancer, you can have a TRAC fusion infantile fibrosarcoma, and maybe that doesn't have to be differentiated. We can think about the different acute and late risks of some of these studies. Obviously, if there's effects on the growing patient, then maybe that's more important when you think about an infant that may have late effects going on. So some of the requirements that we think about accrual and eligibility may change. Formulation, that can also affect your accrual and recruitment. If the drug only comes in a pill, then you've already cut out maybe some of your less than 3-year-olds that are present. So obviously that's important. And we often have these arbitrary age cutoffs. So in adults, a lot of the studies are greater than 18, and pediatrics were less than 21. But why are they there? The median age for phase 1 trials are 12. And so maybe we can think about it more, especially with some of the FDA guidance and regulations that are out there. When we think about patient population as well, again, our numbers get smaller and smaller, so accrual can get harder and harder sometimes. So we cure more patients, which is great, obviously, but we also have less patients that are recurrent and refractory in that setting to enroll on phase 1 studies, which can affect our recruitment accrual. Our populations are getting smaller and smaller too. So, they're being more defined by molecular subtypes in the era of precision medicine. So if I need a fusion negative embryonal rhabdomyosarcoma with an HRAS positivity to enroll in a phase 1 trial, I've already decreased my numbers dramatically by just that small strict criteria. And so that can also affect your recruitment accrual and things to think about. So in the setting of a study structure up here, maybe this cannot be done as a single institution. We can't think about having just single institution trials because you're not going to be able to accrue HRAS positive fusion negative rhabdomyosarcomas with one institution. And so we have to think about, how are we going to include other studies, or do we think about it in a cooperative group? And as Julie said, in a cooperative group, a lot of our metrics are required on accrual. So we belong to the Pediatric Early Phase Clinical Trials Network, which is actually a small consortium within the COG. So it's 21 institutions across the broad spectrum of COG. Our biggest criteria for remaining into this very highly selective and competitive consortium is accrual numbers. And if we don't meet those accrual numbers, it's actually a ding on our metrics. So these are things to think about when you open these studies and who you're going to be enrolling. And obviously the funding and sponsors and how you're going to get the money in terms of opening these studies, who's sponsoring the studies, the travel for patients are things to think about too in terms of recruitment and accrual. If you're the only site, how are you going to, and a patient has to come over to get the drug, that's another thing that will affect your recruitment and accrual. And also there's regulatory restrictions. So, sometimes either your regulatory or your sponsors will say, we cannot accrue this type of patient. It has to be with this type of arm, and that's the only way it will move forward. And that obviously will affect your recruitment accrual as well. And we've had many scenarios such as that where, we've been limited to the patients that accrue, and that can affect, again, how your study runs and goes. So I think in the end, some of the things that I think about is that we really need to think about smart and innovative, inclusive, flexible designs when we think about our studies so that we can include and think about recruitment. It's part of all of our grants. It's part of all the things, how you're going to accrue, what the numbers are, how many patients you see. And that's not only for pediatrics, but across the board. I think that's it for me, and I will stop there.
Terrific. That's very provocative. I'm sure we'll have questions on that. Dr. Chalasani
Yes.
is here. Yes, thank you. Would you like Pesha to show your slides, or are you going to share your slides?
I'm gonna try. My screen was freezing earlier, but I'm gonna try. So
Okay.
let's see. Can you see my screen?
We can.
Okay. I think when I tried to do the slide share, something was happening. So I'm just going to keep it in this mode for now for us to discuss.
Okay.
Oh, sorry. Okay. So I'm just going to briefly go over investigator-initiated trials, and I think it's kind of like a nice segue into what Dr. Kim was mentioning earlier about having a study design and flexibility. And I think that's so key and essential for any kind of trial to be successful. So one of the things that I was going to touch base is investigator-initiated trials, which are always like the crown jewels for any kind of place. And in these goes to all specialties because this is an investigators or institutions IP, so to say, in that sense. So it's your idea that you're trying to take it from A to Z. So it's kind of very exciting in that. So basically, I'm just going to go over like what exactly is a letter of intent and what we go about. Or here it's about what is the investigator-initiated trial, like how do we develop it? So it's the first idea or the concept which can be developed into a full project. So this is generally what I talk to my fellows and the residents and some of the students in like how we are developing, because frequently we are encountering things like how do I go or how do I develop ideas for IITs, right? Like what are the mechanisms? So those are the first ones which come, and it's daunting and challenging thinking, how do I come up with this like randomized phase 3 is like all these things coming up, but it's a stepwise approach and it's a learning process. So the intent is like the first idea, which can be developed into a full project from a preclinical, translational, clinical one. The IITs, like our investigators, like I said, the IITs are the crowning jewels for any institutions because they are the investigators who are involved in the study design, the protocol, the biomarkers, which are, in 2024, trials should not be done with our biomarkers, and endpoints and accruals and everything. Like we are invested pretty much in everything there. So we're kind of briefly going to review about what are the different steps in an LOI, because this is where I think most of us would love some more things. So how do you get ideas, right? The first ones, I always try to remind our fellows and residents and also with the junior faculty is, it's easy when it's not easy, I should say. I think we all get, like I said, it's very daunting when we see these really amazing podium presentations and we are scared to think that that is the first thing we have to achieve, or maybe that is the ballpark, or those are the ones which are visible. So that is by default what we think about how investigator-initiated trials should end up or things, or that is the bar. But that's frequently not the case. So as we are starting out, the question is, what is relevant and exciting for you? So the ideas or concepts for any investigator ideas can come from collaborations, from reading a good article, from mentors. So reading in conferences when you're going through, or these days when anything has been published, we get a highlight or a quick email blast comes with a really high impact paper. So just reading that, and I've actually completed a trial purely based on a paper I've reviewed and has, nothing to do with the mentor or the clinical question. So there are things, just listening to talks and going for conferences, grand rounds, and that is where you listen to speakers presenting good topics, and you pause and think how you can potentially correlate, or is that something that we can take it to the next step? So that's one way people get that. And the other ones are, again, collaborations where you have the strengths of how an institution or investigator can progress is also depends on the collaborator. So lean into the strengths of the division, strengths of the institution, and go and reach across disciplines, go and reach across your divisions, to figure out what the strengths are, who are the strong who expertise in your institution that you can leverage and develop a good concept and going from there. And, obviously the other one would be a mentor. They have a project going on that you can then break out or take a small topic of that and then develop your own projects too. So there are different ways you can develop ideas. There is not one perfect way, but all those things will get us to the next one. It'll start the wheels moving, and I think that'll just start it. Once you get I think we frequently say, once you get bit by the bug, I think in the academic medicine of writing grants and clinical trials, we are hooked onto that. Even though they are not always successful, we just strive to do it better because we see the impact it can have clinically and what it is. So I think that's essential to keep that going. So once we get the ideas and we are like, okay, what do we do next? Usually try to put my thoughts on paper, try to get the background, and knowing what the clinical patient, population might think, like to Dr. Kim's point about feasibility, I meet with the statistician first to actually know what is feasible or not. For me, I've learned from experience also that I don't even finish writing my aims or even figuring out my study design until I meet with a statistician, because I may have this great idea, but it may not be practical or feasible. So I need to really define it based on what I can do, what are all the funding I'm looking at, and things like that. So I feel like I got my thoughts, I have my idea, but it's the statistician who's going to help me put it in terms of a more refined LOI or a synopsis for me to even know what to go from there. And that depends huge on the feasibility. And the next main thing is funding, because everyone is like, okay, you have this idea, but how are you going to do it? And there are funding sources. So that is the critical one. And I think in terms of the funding sources, you can look at internal pilot fundings to see, again, that is where it's critical. Like I said, I don't finalize the IIT or my study design until I have my study station on board and I know what is feasible or practical. And then figuring out, okay, the internal pilot funding mechanisms are coming. Is that something feasible? Do we have to really look for external peer-reviewed funding? Because then you're looking at different ones and looking for those things. Pharma support, sometimes it's drug only, sometimes it's drug plus funding. So looking at different resources. But I also think it's important for everyone to talk to their colleagues and mentors who have done this and who can guide them and saying, okay, this is what you're looking at and going from there. And the last but not the least is the timeline for that. As soon as our idea comes, we want it to be in a trial by next month and start accruing them the following month. But in reality, it takes way longer than that. And I think patience is not an easy thing, but we will learn over time. But it does happen. The main critical piece I only say is it is a long, daunting thing because there are multiple steps in that, but not to give up and not to push it because an idea is worth pushing it. It's just that there's a lot of steps and processes in there, but it is good. And that is also where talking to your mentors and colleagues is very helpful because they can guide and go through that. So like I talked about, once you have the idea, a lot of times things could be done in parallel, like the funding, the budgets, contracts, and writing your IRB protocol. Like Dr. Bauman mentioned earlier about the templates we have available in the cancer center, which could be leveraged and things like that. So there are steps which could kind of help in that. In the ideal setting, everything would go through and you could have a protocol once you get the green light in about 6 months. But best case scenario, but it can take longer and it is okay, but you have the right idea and you should continue to push for that and go from there. But I can tell you that it is very rewarding to see even a small pilot trial, biomarker driven, or whatever your idea was to finish it and see the results, support your hypothesis or other way, but you learn so much and it's really exciting to finish that and go from there. So and that is the reason these are the crowning jewels for any institution, because it's your ideas that you're developing into clinical trials. But the feasibility is the big thing. So always knowing your patient population is the critical one, because that will take your trial, whether it's going to be successful or not, for the next steps. I think that's my last slide. I'm going to stop here.
Okay, fantastic. So I want to open it up to the group to ask our experts any of the questions that you have about accruing to clinical trials, cancer, or more generic other trials. And I think to lead it off, I'm going to ask a sort of softball question from our speakers, which is to tell us a little about their experience. What led to your very first trial? Julie?
So my very first trial was the product of, I went to a scientific seminar. I was in training at the Hutch in Seattle. And I went to a scientific seminar that described a fascinating pathway of overcoming interstitial hypertension in tumors and therefore enhancing drug delivery. And then I studied that pathway and found a drug that could be repurposed to inhibit that pathway. And I developed my first investigator-initiated trial. I actually went to the ASCO AACR Methods workshop, a really great resource, I think in 2006, to develop that trial. I conducted that trial and I published that trial. It took about 6 or 7 years. I did get institutional as well as pharmaceutical support for the drug. And the trial did go from start to finish. And so that was my first, but the kernel of the idea came from sitting in a scientific seminar and thinking about how I could repurpose a drug to tackle that pathway.
Interesting. Erang, would you like to comment?
Yeah, so my first clinical study was actually with the drug sorafenib for pediatric plexiform neurofibromas. And I worked this primarily with the help of my mentor and during fellowship. I was always interested in clinical studies and clinical research, and I actually did a doctoral program in clinical investigation through my fellowship, so this was part of my PhD thesis. But it was actually a lot. I mean, I learned so much on it. I too went to the Vail AACR methods in clinical cancer research, which was, really, and I use this study as that trial as well. So it was actually a very great opportunity for fellows and junior faculty for that who are interested in clinical research. But this trial was also very interesting because I learned a lot about toxicity and accrual. We had no problems accruing because it was for a niche population, and I was at the NCI. And one thing about the NCI pediatric oncology branch is that it was a referral center. So in terms of what the NCI provides is that it provides travel, allows people to come. And so you can sometimes run single institution trials out of the NCI because it refers across the nation. But it also learned a lot about formulation because, and this is why sometimes it's very difficult to run a phase 1/2 trial as a fellow when you only have 2 to 3 years to finish. But the trial was on hold because of formulation. So there was a 50 milligram tablet for sorafenib that I needed for my pediatric population. And at that time, Bayer was supplying the drug, but it was behind production. And so it took almost 2 years for that tablet to be made so that we can create the nomogram for our pediatric population. So I think those are some of the trials and tribulations when you think about creating trials and designs, especially for a niche or a rare population and in young cancer patients or young pediatric patients. But we did finish the trial. Unfortunately, it was too toxic for our patient population. So it did not move forward in terms of a drug, but I actually was able to take that and open the sorafenib trial within the COG phase 2 component as the study chair for that. So it led off to other things at the same time.
Thank you. I noticed that Pesha has put the link to the ASCO AACR methods course that's in Vail this summer for all of you who are thinking about contributing to that workshop. Pavani, would you like to reflect on what your first trial was?
Yeah, and I think in lines with what Dr. Bauman and Dr. Kim mentioned, I think most of us, I think ASER, the Whale workshop, is our first dibs at like writing our own IIT. So that's similar for me too. So when I was a fellow, that was my first, I would say, where I got bitten by the biomarker bug is like my trial. Like most of the trials at Whale were therapeutic trials looking at novel mechanism. And I think mine was a biomarker-driven trial, just looking at a new one. So it was at that point my mentor suggesting, Dr. Livingston suggesting it. We had an idea about DNA repair, and this was before we have now the methylation and the BRCA nest-like concept in a lot of tumors. So we were looking at tumors which would have a higher DNA repair deficiency for reasons other than BRCA1 or 2 mutations, kind of like categorizing them in the BRCA nest and treating the patients with DNA damaging drugs like doxorubicin in the metastatic setting with the hypothesis that they would be more responsive to anthracycline DNA damaging drugs. The trial was not completed successfully and we were not able to do, but that's also a lot of learning in terms of how biomarkers are designing. To Kim's point, we learn a lot about those nuances and what takes the biomarker development, how the tissues have to be stayed, how we have to get access, like how do we store them and how do we process them, like things that I've never ever imagined or ever thought about it. Just look at the path report all the time, but don't actually think through those processes. But those are critical and essential, and because they directly link to the treatments and the outcomes. So I learned a lot. We were working in collaboration with Ventana, which was later got on by Roche. because they were based off in Arizona, in Tucson too. So we had a good collaboration in developing the assay, and we presented an abstract at San Antonio, with some of our preliminary results. So we closed the trial because we were not able to successfully transfer our developed assay in tissues like archival tissues. It's just not optimal. So we couldn't I would say, finalize our results and present it, but it was a valuable one. It was a biomarker-driven trial at that time.
Terrific. I don't know all of the names of the people who are on the talk today. I don't know how many of you have ever participated in a clinical trial. If you have, would you go down to the reactions tab in the bottom and give us a thumbs up? Okay, it looks like not many people among the folks that, yes, Naomi. Thank you. The buttons work. So I guess that if we're thinking about folks who are considering clinical trials as part of their career future, I wonder if someone could explain this business that all of you mentioned, which is cooperative groups. How would one of the folks on this call today get involved in a cooperative group? Is that through a mentor? Is that through an institution? Is that through a specific trial?
Well, specifically for PEDS, I think all of the above actually. So all of our clinical oncology fellows, hem-onc fellows, are members of COG automatically when they become fellows. But there are other consortia as well, not just COG. Obviously, that's our largest one, but like I participate in the DoD and NEV Clinical Trials Consortium, the Sarcoma Alliance for Research Through Collaboration Consortium, and many of these I've been brought through and have open studies within these consortia through initially through introduction via mentors, and now luckily I'm getting the chance to do the same for my mentees as well. So I think there are many different ways to participate. A lot of these consortia also have young investigators groups that one can join as well, and then even be assigned to a mentorship program.
Yeah, I think that's critical for the cooperative groups. I mean, they do have meetings, and they have more, which are different from the major national meetings. I think what I've learned from experience is, like, it's critical to show up for those meetings because they're smaller groups. And I think, because like Dr. Kim was mentioning, a lot of times when you see new faculty or investigators and they show up and they show interest and message, as protocols are being reviewed and they are assigned, I think there's a edict, and the NCA really wants to bring in new investigators and early investigators. I hate to say it, but a lot of times it's like the people who are there, they make decisions sometimes in real time, and they see you and they're like, oh, okay, we'll get you in and protocol. So is showing up and being present at those meetings, I think, at least for cooperative groups, is a critical part.
Right. And my entree into a cooperative group was and beginning to have leadership and the opportunity to develop trials within the cooperative groups was also related to showing up and opening cooperative group studies and accruing to them. So, becoming an accrual author is an incredible entree to being viewed as valuable. And then people who are good accruers to a concept often are then invited to be the co-chair of, a new trial that comes along in that specific working group. So showing up, joining the new investigator committee, proposing things. I think they used to joke at ECOG that it wasn't an ECOG unless Bauman presented a new concept. And accruing to show the value of that cooperative group to your practice.
Good, excellent. Naomi, there's a question in here about examples of successful grants in the grant library, and I mentioned the Grants Enhancement Program, and there was a follow-up question that says the person's curious about lipid liquid biopsy assays like plasma DNA, RNA, molecular biomarkers for pediatric solid tumors. I know that's an area of real interest at Children's.
Sure, let me take the first one and tell you a little bit about the second one, and I think Arong may be able to add to that. So yes, at Children's, we do have a library of successful grants of different kinds. They are under the oversight of the Grants Enhancement Program. The person that you should get in touch with is Dr. Stefan Ladish, who can give you access to those grants, and who also is available, in particular, available to you, Stephanie, to help you with grant revision, specific aims oversight, and to provide those elements of detail that everybody needs for the front or the back part of the grant. Apropos of liquid biopsy assays, that's sort of an area that pathology and our research pathology, put that in quotes because we really don't have research pathology, is just now getting interested in. And there are 2 new hires. I happen to know that because I've reviewed their CVs. One of whom has for sure signed on, and another one we hope will sign on, who will be bringing liquid biopsy writ large to the institution. And Arong might even have a little bit more detail than what I just said.
So, I mean, in terms of like what we do for clinical studies, I think it's a very important, we don't have, a lot of liquid biopsies we're learning more about now, especially in the pediatric population, and the usefulness of what we're looking for, and the different histologies that may be related to. And I think the most important thing right now that we're doing is collecting the samples. So within all of our COG studies, they are collected in various portions for our early phase studies. For the upfront sarcoma studies, you can see that there's a Streck tube, and you're collecting them at specific time points. And for the studies, and particularly the MPNST studies that I write and participate in, we are really trying to figure out how to best collect it, what time to collect it, who are the collaborators that are going to be that collects it. But I think most importantly, we have to think about it and incorporate it into our studies, and that includes liquid biopsies and other biomarkers of interest that we want to do, and think about it in the time point of when we're going to collect them. And again, thinking about our patients, how much we're going to need, when we're going to do it, how do they correlate with imaging and response, and many things like that when you're thinking about the study design.
In the real world, it gets to be quite interesting because you might need to collect samples to go to the cooperative study, because that's part of the criteria of participating in the study. And then you as a single investigator might want some samples. And so the whole issue of how one is consenting for all of those different things, for a cooperative study sampling, for an individual investigator sampling, for a biorepository, because you never know what's going to happen in 6 months, a year, or 2 years, sort of becomes a really, really interesting subject that we don't have time to discuss today. But apropos of that, one of the things I thought, unless there are more questions coming, we might talk about is single IRBs. Because you need a single IRB if you're part of a cooperative study. And if you're an independent investigator joining with 3 or 4 local investigators, and you're NIH or DOD funded, you need a single IRB. So how many of you know about single IRBs? And if you don't, that's fine, because we have 2 experts on with us today who can help you.
I think that should be delved into rather more fully in another discussion. It is a serious issue, no question about it. But that kind of leads me to a more general question, which is, we've been talking about participating in cooperative groups or perhaps in multi-site trials. And I guess I wonder if you could sort of speculate as to how an individual investigator gets sufficient credit when participating in one of these larger groups. We're at an academic medical center, you're part of a big trial, you're going to be number 47 on the author list of the paper eventually. How do you feel you get sufficient recognition of your efforts in part of a larger group? Are there strategies that will increase your participation or visibility or rewards?
Is this to the panelists, or are you asking
I'm asking Julie.
asking me? So it's a big question, right? Like, so how do we actually recognize team science? And I think there's a number of different ways that it can be recognized. Thinking first about the Cancer Center, right? So clinical trial accrual is a multidisciplinary event. Hopefully, a patient is presented at a tumor board, and the group consensus, because clinical trials in cancer are frequently conducted in the context of standard of care. So we introduce ourselves to patients and we're talking about what's standard of care and what's the clinical trial option for you. But so the Cancer Center, for example, when we have an accrual and that accrual was multidisciplinary in nature, we give accrual credit to both the surgeon and the medical oncologist, say. So that's internal. That's a way of recognizing, as we build a cancer center and build the metrics, at least in adult oncology, of what true contribution to our collective goal of building out our clinical research for our patients. That's one of the ways that the Cancer Center does it. In addition, thinking about, earning accrual authorship. So I think that's one of the really important things that you need to consider from a feasibility standpoint, is once you understand your population, opening trials where you can hit it out of the park, where you can earn accrual authorship. Getting 1 or 2 in is not going to earn you accrual authorship. It will potentially, if you have, the portfolio open and you're showing up to team meetings, it will be recognized that you have put, say, 10 patients on ECOG trials that year. However, to earn that accrual authorship requires that you have a certain proportion of the patients that you accrue at your center. And that is possible. That's in fact how I earned my first cooperative group authorship, was by choosing a trial that I felt was a really good match for my population and offering it to every single person who met criteria. And I think I had only 1 person say no. I think it's also about, I think that those are the main things. I think we should do additional, reflection on how do we reward team science, because that is absolutely critical in clinical research. Yes, one other thing I would say is that investigator-initiated trials, especially if the spark for them is initiated by the science that you hear from a colleague or a collaborator or the research program. Investigator-initiated trials are the perfect specific aim 3 for a collaborative R01 grant, because you can build into that grant the human experiment of, the biomarker that you embedded in that clinical trial, and the resources for studying that biomarker, that mechanism are embedded within specific aim 3. So for the longest time, I've called myself the specific aim 3 person. So the clinical trialist, really, I'm an obligatory team scientist. And in that way, one develops the ability to have an investigator-initiated trial where it's yours, and you will be the first or senior author of the trial itself, and then a co or a lead author on multiple publications that stem from that grant.
Yeah, and I think, just to add on to what Dr. Bauman was saying, it leads to additional opportunities. So when you participate in these studies, either if it's through consortia or even industry, and you meet with other collaborators in that, it leads to additional opportunities. So maybe you're interested that drug, and you want to use that drug for your for your own study for maybe a different mechanism that gets you away in terms of working in terms of collaborations, or maybe you're interested in working with this group. And so I think for me, I was interested in early phase studies. I opened phase studies. I learned about them. I became, a member of like a role in COG. And then I got to be the Pepsi TN institutional PI for that. And now I work with other and then you can lead some of the studies within those cooperative groups because you started off in participating them. Or I can use these drugs to think about my IITs that I might be thinking about for other studies. So I think they lead to other opportunities within, and so and that way that develops your academic portfolio in a way.
Yes, Heather?
Hi, I kind of piggybacking on the discussion about working with a group. Do you guys have any tips, like pro tips or pitfalls that we should avoid if we are working at a single institution and we're worried about accrual and we'd like to reach out to investigators at other institutions to try to expand our recruitment? Any like special things that we should know about?
I think like the funding. So in order to open, like if you have a study, you need to have funding in order to support those studies. So I had a trial open that was a single institution and we needed more accrual. So we reached out and we had to think about geographic locations. So is it far enough from us that they can have a different catchment area? How much money can we support per patient with the funding that we have? And how and one of the things I had was a specialized instrument. So what other site had that instrument and will that have that population? So I think those are some of the considerations that we had, but for sure, there are times when, inviting other studies, other institutions to be part of your study is a way to increase your accrual. But it does take a lot of time and effort, especially if you're the coordinating center. And you have to get there. It takes a lot of time and effort and funding. So it has to be worth it in order to do so.
Right. I would say that, transitioning a single site IIT to a multi-site IIT, I've been in those shoes and it is challenging, even if funded, because of the issue of the double indirects, right? So any funds that you send to another center will also be taxed by that new institution. So I guess one of my pearls, but sometimes it's not feasible, is to write it as a multi-site center study from the beginning, with a funding plan that accounts for some of those issues. And sometimes people will do IITs, often people do IITs as a labor of love, provided that minimum costs are covered. So don't be shy about trying, because even a poorly funded IIT, especially in the eyes of a cancer center, for example, has much more valence than something that's industry or something that's a Me Too study.
Thank you. Those were helpful.
Other questions from the group? Please take some time to evaluate this webinar. Do you want more of this? Do you want less of this? Or do you want a different subject? We take those surveys pretty seriously. It's in the chat for the link. So if you wouldn't mind opening that. I'm sure that our resident experts would be available by email or other forms if you need to chat further with them about some of your ideas that you may not have had a chance to talk about today. I want to thank everyone for coming today. I particularly want to thank our cancer accruers for their pearls. We didn't discuss too many of the pitfalls, although they hinted at a couple. And there's more to this story, I'm sure. I want to thank all of you for attending, and please give us an evaluation if you'd like to have more. So I appreciate it. Thank you so much. This will be put on a recording, and we'll send you all where the location of all of the special interest groups are located. Thank you so much.
Thank you.
Bye-bye.
Bye.